This is the image biomarker session. I think it's something we're all sort of looking forward to hearing. This session, I'm co-moderating with Bruce Pyenson, senior actuary from Milliman, former consultant to the Medicare. And this session is going to start with a short talk by Dr. Leest, who is here. Oh, yeah. Okay. Come. Is yours? Do we have your slides? How long?
These,
It should be.
yeah. Yeah, hi everyone. My name is Paul van der Leest. I'm a postdoctoral researcher at the Department of Laboratory Medicine in the Netherlands Cancer Institute. Okay, so although this session is about imaging, of course, I was asked to give an update on the position of liquid biopsy testing in lung cancer diagnostics. All right, so if you look at the events during cancer development, then of course, when you have a low tumor fraction, you can maybe identify that using screening or whatever. And then you have a local intervention. And after that, you can follow up the patient and then you hope the recurrence wouldn't develop. But if so, then from that point on, you have to profile your tumor molecularly so you can guide systemic treatments. And then you can monitor the disease development for response. And if progression occurs at one point, you want to know what the acquired resistance mechanism is there. So you can have maybe a different treatment strategy. So if you break that down in diagnostic terms, then you have in early stage screening and surveillance. And in the advanced stage setting, you have primary diagnosis of the tumor, treatment response monitoring, and acquired resistance detection. And now currently we're doing that in the early stage, usually based on imaging. And in the advanced setting for primary diagnostics, we're usually using molecular profiling of tumor tissue. For response monitoring, we're using imaging again. And then for acquired resistance detection, we're back to tumor tissue. And now today, I will try to show some initiatives that we had to see whether liquid biopsy testing would also be beneficial in these settings. So liquid biopsy is basically just an analysis of tumor-derived material in the circulation. And you have different biomarkers in a blood sample. You can look into circulating tumor DNA, circulating tumor cells, circulating RNA, extracellular vesicles, et cetera, et cetera. There are a lot of different factors to look into. And for today, I will only focus on circulating tumor DNA. So circulating tumor DNA is kind of challenging to detect because it's actively degraded in circulation, which results in a very short half-life. It is fragmented material, so you don't have long stretches of DNA to work with. The abundance of circulating tumor DNA is relatively low. I mean, the DNA that is derived from the tumor is only a small fraction of the total circulating DNA there is. And most of the DNA is from the hematopoietic lineage. So, and also some other factors, like physical factors or whatever, can easily contaminate your DNA fraction that you have in your circulation. And also during the processing of blood tube, there are still some events that can occur to induce hemolysis. So altogether, to be able to work with ctDNA, very ultra-sensitive methods are required. So if you look at ctDNA as a biomarker, there are basically two things that you can do. Either you can use it to characterize a tumor, or you can determine a tumor fraction, basically a level of the amount of ctDNA that you have. So for molecular profiling, we're relying on the same molecular markers that we have in the tumor, which would be SNVs, indels, fusions, or CNVs. But if you're looking at only the determination of a tumor fraction, then you have multiple things you can look into besides of that. You can look at somatic focal amplifications. You can look at methylation patterns, fragmentomics, et cetera, et cetera. So you have more biomarkers to work with. So if we look at the early stage for what the beneficial effect would be of liquid biopsy testing is to determine a tumor fraction based on ctDNA abundance. So we have some challenges in early detection. We know from the Nelson study that the positive predictive value of image-based screening is around 44%. For ctDNA, we also have some issues. That's basically that the sensitivity drops when you go to earlier stages of disease. So for stage 4, we have over 90% concordance with tumor tissue, while if we go to stage 1, it's approximately 60%. And also finding the appropriate analysis method is also kind of challenging because you want to have the ability to detect as many cancer types as possible, so irrespective of the histology and the molecular pattern in that tumor, but you still want to remain a high specificity and a low false positive rate. So the question is, how do you integrate ctDNA testing to improve image-based screening? So if you look at the both markers, you have screening on one side and our molecular biomarker on the other side. And you can, of course, use that for risk stratification of individuals, which you want to follow up afterwards. One possible way would be to have a molecular confirmation based on your ctDNA from screen positive individuals. So we first screen everybody. If they render positive, you check for ctDNA. And if that's positive as well, you have a double confirmation. You can also do it the other way around. You just test all the blood samples, all of the patients, and when a ctDNA positive sample comes up, then you, after that, would have a personalized imaging strategy for that individual. That also, of course, depends on what sensitivity you can reach, whether you would like to rely on the one approach or the other. I also want to highlight a surveillance strategy that we're assessing at the moment in the Yankee. But it's not in lung cancer, it's in melanoma. But in melanoma, the current standard of care is to resect the tumor and then have radiological follow-up over time. And if the radiology shows that there is a recurrence, then the patients are treated. Here we're looking now in a retrospective study whether when we add ctDNA testing to that follow-up, whether we can see what kind of growth patterns are common in these individuals. So we can basically expect what the range of an earlier intervention would be based on ctDNA detection instead of imaging, with the ultimate goal that if we, for example, apply this in the routine, that we can, based on the detection of circulating tumor DNA, already inform the clinicians that something is about to happen, so they know when to start the imaging process, and then have maybe an earlier intervention possible. And after that, we would like to maybe do an interventional study in which we can see whether we can reduce the imaging standard at this moment and only be guided based on the circulating tumor DNA. And then afterwards, when that renders positive, we have an imaging. So then we go to advanced stage disease. For primary diagnostics and acquired resistance detection, we basically want to see what the correlation is between tumor tissue and liquid biopsy. So can I retrieve all the mutations that I see in the tumor in circulation? And if we look at response monitoring, we want to see again what the abundance is of ctDNA so we can guide the imaging. So if you look at the concordance between tumor tissue and plasma, we see that in 82% of the patients, we have a similar result in the tumor compared with the liquid biopsy testing that we did. And if you look specifically at the actionable mutation, so the mutation that you would use to guide treatment decisions, there you see that you have approximately the same number of cases that you would identify based on your liquid biopsy testing as you would have done on tumor tissue. So this actually shows that you can use it as a clinical application for treatment guidance. And if we look at response monitoring, we can see that if you have 2 different intervals, for example, at baseline and a couple of weeks into treatment, that you can stratify patients based on ctDNA decrease or the absence of a decrease. Which has a very beneficial effect on the progression-free survival and the overall survival of these patients. These are all lung cancer patients, by the way. And also, if you monitor longitudinally during the treatment, you can see that a complete response, partial response, stable disease, or progressive disease, so basically the RECIST criteria, correlate with the ctDNA levels that we identified. So if I show you all this data, then the question arises, why is liquid biopsy testing not implemented yet? Well, there are several reasons, and I'll highlight a couple of them here. There's an overwhelming availability of commercial products. So there is a lot of different blood collection tubes, cfDNA extraction methods, reference standard, and analytical applications, which really make it difficult to compare and show enough proof that you can use this in a routine. There's also a lack of harmonized standard operating procedures, so you cannot really compare the one data to the other. And there's limited possibilities of reimbursement as a consequence of that. So which considerations in ctDNA testing are crucial at the moment? So we wrote a review recently about this. It's published in Clinical Chemistry, in which we highlight a lot of the pre-analytical considerations, but also the analytical things that you have to consider to start up with liquid biopsy testing. And I'll highlight a couple of them here. The extraction method, we showed that if you just compare a lot of different labs within the Netherlands, that you see a broad range of the yield of cfDNA and ctDNA based on the methods that they use. If you look at the analysis method, you can also see that within the Netherlands, there are a lot of labs using different methodologies, and that has an effect on the number of mutations that you can retrieve during your diagnostic follow-up. And you also have to consider that the mutations that you're testing for are really derived from the tumor. That's also why it's important that there's a lot of the DNA in the circulation derives from clonal hematopoiesis. Sorry, from blood cells. So clonal hematopoiesis can be a contaminant in this factor. So jumping to my conclusions, ctDNA is a promising biomarker for lung cancer diagnostics. Although accurate ctDNA interpretation is key to prevent misdiagnosis, there are a lot of discrepancies still in the routine in pre-analytical and analytical workflows that prevent the implementation of ctDNA testing at the moment. And therefore, there's a demand of EQAs and prospective clinical studies to show the clinical utility of liquid biopsy testing.
So this panel discussion is going to focus primarily on image biomarkers with a view towards indeterminate nodules. And we have a wonderful panel here, primarily image, but we do have one blood guy stuck in there who's surrounded on all sides. So I'm just warning you. So, but first, I'd like each of the panelists to introduce themselves and just perhaps say what your role is and what your product is meant to do. Frederick, please start.
I'm Frederick Bragg. I'm the CEO and founder of a company called Median Technologies that I started around 20 years ago, where the whole idea was how to apply technology to identify imaging biomarker. It's a huge need for non-invasive marker. You basically have two, liquid biopsy and imaging. I tend to believe, just like for the presentation, that it's a huge of common ground that can happen and that the two might be used in conjunction. But and we have two sides of the business. So we develop software as medical devices, which is a huge endeavor. It's like going through a drug process to take it to market. And we're currently going through our pivotal study for lung cancer screening, which is both to detect potential cancer, but to be able to characterize it. So looking at images and be able to say, is this malignant or not? And we have another side of the business where we work with big pharma, applying imaging marker. It could be simple resist, but much more advanced markers to try to help them develop better drugs. We currently managing around 300 clinical trials for big pharma, 90 of them in lung cancer. The average response rate is around 15%, so 1.5, it's really low. And for those who do respond, within 18 months, they do not respond anymore. So it's trying to identify, what biomarker could we identify? Could we identify EGFR mutation using imaging? Because of course, just like for liquid biopsy, it will avoid the need to do, biopsies on millions of people, try to understand when they have advanced disease, what sort of marker could be helpful for them. I will not take all the time, but just one thing I want to say, I feel incredibly honored and humbled to be here today because what all of you have done is something that could be, to my mind, the single largest impact ever on cancer care. We know it very well at Median, where we handle the 300 clinical trials for drugs for patients who unfortunately will die. Here, you have proven something that is absolutely crucial. Find it early. It's a direct correlation between the stage of the disease and the capacity to save the patients. And I found remarkable all the panels we had this morning to see the challenges, even though you have proven this, the time it takes to really implement it and really make it happen. And I have a zillion opinions on that, but that would be, I'll let the others speak too.
My name is Janshna Devi. I come from India and we are doing Kyoto. It's an 8-year-old company where we started as how do we support TB screening, how do we really cost effectively just exist. And we ended up understanding the module and module management. Where on the chest X-ray, today we are at a place where we have processed about 2.5 million scans. And were able to alert and flag with the LMICs in developing countries, including India, African regions, and Vietnam, where we were able to flag 60,000 high-risk patients on the justice who enter into the lung cancer pathway early on. Because typically justices are taken from younger and healthier population. So that's been working on care all about. I have extensively been working on with the R&D and the AI scientists. I really to understand how a nodule not just stop at detection, but the biomarker side of the medical imaging on chest X-ray and on the chest CT to a level where not just thinking these two are independent modalities, but behavior of chest CT can be projected on the X-rays itself as if the AI models can fix certain biomarkers using CT as a gold standard, right? I think the interesting opportunity right now, what I can see is that as liquid biopsy with fluid biomarkers, blood biomarkers become more and more relevant. We will essentially end up having a lot of data around the blood biomarkers, giving us molecular level information, which we could then see how this information for the same patient, if they have images taken, could we come up with the same level of data and analysis to see if these can be picked up on the imaging analysis, the medical images itself, or together with image blood biomarkers, we can create a whole way of saying whether or not it is cancer or not.
My name is Luis Montuenga, and I'm very strange to speak here honestly. I'm a translational scientist. I work on biomarkers in liquid, but not only liquid, but actually tissue-based biomarkers in lung cancer, especially in early stage lung cancer, and especially in screening. I've been working with in the I-ELCAP group in Pamplona and the Navarra, which are the Madrid side of the University of Navarra, the main campus is in Pamplona, in northern Spain. And we have had already two I-ELCAP meetings there, many years ago. And so I've been working on biomarkers in the context of screening of lung cancer, where within the I-ELCAP group of the University of Navarra, where the Italian and the group of one of the models much more. Anyway, and my job here is to convince you. Well, we have had a great introduction by Paul, and from apart from imaging, also liquid biopsy and other biomarkers may help in the aim of risk, in this case, not characterization. And I'm a believer in liquid biopsy, but also in imaging. So I'm not counteracting any of the arguments that we've been here, but I think what you just mentioned, collaboration, both contributions are complementary and can
Can you hear me? Yeah. I'm Václav from Optelum, founder of Optelum in Oxford. Our vision for the past couple of years has been to enable every patient with lung cancer to be diagnosed early and treated in the optimal, ideally curative way. I guess why most people know us, we are the only company who introduced an FDA cleared and reimbursable for US Medicare patients. That's the biggest healthcare payer for 100 million Americans. AI decision support system for early stage lung cancer, which effectively helps hospitals automate setting up a nodule clinic program. So it leverages patient discovery based on natural language processing and radiomics to capture at-risk patients from any type of scans and then accelerate the right ones into a more aggressive or less aggressive diagnostic process. What we are doing going forward is extending into precision treatment. If some of you attend ASCO in Chicago in a couple of weeks, so there we present first results with Professor David Carbone in extending the work into guiding neoadjuvant perioperative treatments with through AI augmented recurrence prediction, showing double the sensitivity compared to clinical staging.
Great. I'm Michael Calhoun, founder and CTO of Reveal DX. And a big part of what we're doing is quite similar to what Václav and his group have been doing. So I want to congratulate him on their progress. But our entire company right now is focused on a very simple and I think very important question, which is when you find a lung nodule at chest CT, is it cancer or is it not? And what we're talking about, obviously, in very simple terms, can the circulating biomarkers do that? How are we doing right now with the guidelines? And with the guidelines, half of the cancers are found when they're under 8 millimeters. So half of the cancers, because of how frequent the small nodules are then triaged to follow up, come back in 6 months, come back in a year. Can we do better at identifying which of those nodules are cancer? And the answer in our published evidence is yes, we can do that. And there are more papers coming out. The important part of that is that we understand, like has been talked about, this fits in to end-to-end patient management and clinical management. So we're working with companies like ContextFlow, their CEO is here, and Mavis is here, so that we have a comprehensive radiology package where the nodules are automatically detected, the risk is stratified, and then that information can be passed downstream to the clinic. I'm really inspired by Claudia and David and the I-ELCAP team, and the sun is rising this morning. That was partially because, in my view, a year and a half ago, I heard the word cure talked about in lung cancer, and they're showing for long-term survival, over 80% lung cancer survival, when the US national average is 27%. So then the question is, what can we do better? And that is triage more of those early patients with certainty into early action. I really appreciate what you guys have done.
Okay, can you hear me? Yeah. Okay, my name is Jang from South Korea Core Line, and I'm a medical doctor as well as an AI researcher. And our company is a chest CT company that deals with nodule management as well as detection. So our product is basically CAT-E program, as well as we are doing a lot of collaborative research with European clinical trials and Korean researchers. And our point of view is that, of course, we have to diagnose and manage the nodules themselves, but we are focusing on medical doctors, clinicians' decision. So we think that just giving them the malignant score or some kind of malignancy prediction can be a little bit different from our point of view. So we're focusing on CAT-E program. So yeah, we're doing such things.
Afternoon. Yeah, I suppose you can hear me. So my name is Faiza Admiral Behlul. I work at Deep Health. I'm the Chief Product Officer for the Clinical AI Lung Solutions. It might sound like Deep Health is a new kid in the block. Maybe you know some of our solution because they're coming from Aidence. Aidence was acquired by RadNet. RadNet is one of the largest radiology service provider in the United States and has this vision of bringing AI to their premises, but also make it available for anybody else who could benefit from their experience, but also for the AI, from the AI in general. So our products today are mainly based on what Aidence had for long, are mainly based on what Aidence PV, as you know it, has been offering. So we are in the CAD-E domain, but also the characterization, and we also provide nodule clinic solution, mainly for the incidental, but could be potentially used also for screening. As Deep Health, what we're working on is beyond the point AI. So basically, of course, AI is powering and enabling a lot of things, but what we have learned is you need a little bit more to help the adoption of AI, and AI can be applied beyond the imaging biomarkers. But because today is only the imaging biomarkers, I'm not going to tell you what we are doing. But as part of adopting AI, I think there is much more to do on along the care continuum, or at least the screening care continuum for the lung cancer in particular, but also other cancers like breast or prostate. So I invite you to contact me if you'd like to learn a little bit more beyond the imaging biomarkers. But for the sake of this panel, I'm going to stop here, and looking forward to a good discussion. And thank you for having us. It's a pleasure.
Thank you. Okay, so as you can see, all of these folks here are working on similar products, or there's certainly a lot of overlap. And what we're not going to do is ask them to compare their performance scores and say, I have an AUC of this and my sensitivity and false positive is that. I don't want that. What I'm trying to do is draw out some general concepts and principles and see what the overall direction these companies want to take and why. So I have sent them a list of questions. I hope all of you had seen some of them, none of them. We did send them, I promise. I think you got to talk to your staff.
Oh, we have some that we didn't send.
But we'll come up with some news. But here's the first question that we have is in the domain of an indeterminate nodule. How good does your product, any of your products, really need to be to make a change? I mean, what you're trying to do is change practice, right? Eventually, if you do nothing, whatever it is going to declare itself, right? So as best I can see, all of these products, whether it's a blood product or an image product, is going to shorten the time, the diagnosis, right? So how good do you have to be so that you sort of do that? It seems to me it's a balance of, we can wait, maybe we'll wait 6 months, but if we do it today, we don't have to wait 6 months. So that's where your balance is, right? If you wait 6 months, something is potentially lost if it's cancer. And if you do it today and you get the correct answer, you save that 6 months. So that's where the balance in our mind is. So how good do you have to be? What type of performance levels are you thinking of for the products to really be useful? Is this a reasonable idea? Does anybody want to take that first?
I think that's the fundamental question, right, David? You've been an expert in talking about what are the factors that should be used in order to follow up now or not. I think I would add one thing to the question, which is it also not beneficial to not to have to do that CT scan? So there's economic benefit of that, and there's certainly patient worry and patient time benefit of not doing those extra procedures. So I think if you look at the metrics, the KPIs that were put on the screen earlier today, it really is making decisions for follow-up care. If you're better than the current standard of care, which is guidelines, and you're better than the radiologist performance on their own, then you should be implemented, because it is going to diagnose early and thus stage shift and potentially save lives. And you need to be able at the same time not to increase the unnecessary procedures, at least not increase them, if not do better.
Right. Does anybody else want to add to that answer? Not everybody has to answer this question.
I would maybe add it depends used by whom. And I think if you take one of the world's best thoracic radiologists like David Yankelevitz, probably the bar is too high. But most patients, and Claudia, most patients are managed out in the community by generalists, general radiologists, surgeons who are not specialized in lung cancer. And I think that's where the AI can actually add huge value to these non-experts and basically bring them, accelerate them and bring them to experts faster.
So the idea of sort of leveling the playing field, bringing it across. Any,
But improve the specificity of the imaging, the state-of-the-art imaging is very important, and because everybody is looking at that, and try to reduce the unnecessary procedures, etc. So that's an indicator, and you can use to measure the fix of your device or your imaging or radionics or your liquid biopsy biomarker. So there is something that I think it's important. You cannot, for example, do lots of imaging, sequential imaging on given patient. So that's something that you have to avoid. So you get more specific indicate in the case of blood-based biomarker, this is an advantage. You can take blood from the time.
well, two things can go wrong, right? You can say something is benign and it was malignant, and now you've waited potentially a year for something to happen, right? A malignant tumor is going to go. So we have to know how often you might call a malignancy benign and what's an acceptable rate. And the opposite, how often might you call something that's benign malignant and you go straight to surgery or you get a biopsy? And those seem to be, and then of course there's that whole sort of category where you kind of say maybe it's malignant, maybe, it's in that middle category, and then you haven't added anything. You're still going to get the follow-up CT, right? So do you have any ideas about what's an acceptable number of cases or proportion of cases, or what metrics do you like to think of when you start to say, we can't be calling malignant nodules benign? What's the frequency or that can occur that would be acceptable for a product? Is that something you think of? Is that a way you think about your product?
It is certainly a way we think about our products, but I think the question that this question we are always also asking internally, when is it good enough?
Yeah.
But this is not something that we can define, because we are developing tools that are going to empower you. And we know that there is no test that is 100%. And we are putting AI in a level, it's new. I think the biggest challenge is how can we build trust, even if in a system that is not 100%? Because what we are seeing is, if we bring a solution that is 100% sensitivity, 100%, it will be always based on a cohort, whatever we do, unless we train on everybody. So I think we need to work on what is good enough, knowing that it's not going to be 100%, but that is something we have to work on together. And then how can we build trust is actually the biggest question that we ask internally, at least at my team. How can we make you feel comfortable using AI, even if it's not 100%?
Right. Well, that's sort of what if I were to sort of distill what you're saying, is that you want to know that you're performing at least as well as clinical care.
The beauty about the products that we create with AI is that, let's say, a hypothetical hospital or a clinic that is very good at saying, when they look at a model, they're very confident in saying it's cancerous. But they also fail to say whether the benign model, they're not very accurate. And that's where the problem to solve for them is, how accurately can we follow from rule out the benign models. So in AI, what happens is you can adjust the thresholds or the operating points to make sure that it is customizable to the client or the site on what solution would they actually face a problem. Ultimately, two problems that I see, especially with the intermediate bucket, is that you want to help clinicians to, when they look at an intermediate model, to rule out the models as benign as with a high specificity possible. And if with AI technology they can rule in a cancerous model with lower rates. Both of these things are possible with AI with an adjustment layer threshold. So where is the side failing and how can AI play? So I think it could also be as how the site would want to play with the product as, and I think
Yeah, actually, many of you might have known, but there is a company, AI company, that has a high AUC about diabetic retinopathy, about 99%. But there is another company that has a AUC around 0.8 or 9. But the truth is that the lower AUC company is used in real clinical settings. So what I want to say is that we should not focus on the numbers. We should not focus on the AUC or accuracy or false positive numbers. But I think that medicine is a system, not a technology. So we have to focus on what product can be used in what clinical settings. So I think high sensitivity model, high specificity model, that they are all valuable. And depending on the situation, we can choose whatever we want. So I want to say that we should not strongly focus on the numbers.
Or I suppose we could pick 3 or 4 of them for any one patient, which might raise some of the challenges with regulators and the system. I've got a business model question. I've got to start with a little bit of a story about the way the wine industry works. And I'm pretty sure it works this way around the world, but this is a US story. So the wine, the company, the organization, the vineyard that makes the wine might get paid $5 for their bottle. If you go to a wine store, they sell that bottle for $10. And if you go to a restaurant, they sell that for $20, which is about the most I would spend. But and I understand, that's actually the model for laboratory tests in the US with the originator company. Not talking about things like CBCs and the panels that Quest or the big lab companies run. So that's a kind of an interesting model. It's perhaps more complicated for imaging, how that if it even has been worked out. But I'd like the panel's response to the business model. What sort of value in that proposition? The companies here are sort of, you're the vineyards bottling the wine, and it ultimately gets priced up to what a payer pays. What value do you see bringing in that business model, and as a add-on to imaging and an imaging software, or as a standalone test? And does anyone want to take?
I think that's an absolutely crucial question you're asking there as well. And if you look at it, for years, imaging have been the poor people of the whole Medicare reimbursement system. It's AI has never been reimbursed. So now for the first time, thanks to people like Optelum who've done good work moving that forward, you start to have a reimbursement. And I think this is the whole US healthcare system is driven by reimbursement. So this is going to really help deploy something that can be standardized, used by everyone in the clinics, and provide huge advantages, hopefully, in saving patients' lives. So, and to the previous question David had as well, is if you can prove you have a state shift, of course, it has a huge impact. And the beauty, that's the question you had is true for any diagnostic system, actually. So, what should be the threshold? But it's been absolutely, even lung cancer screening has the largest impact on lung cancer ever. You will reimburse a drug 100K per year per patient, and it took years to try to reimburse lung cancer screening. And the reimbursement, I think, is around $240 today. Makes it hardly profitable for centers to do a lung cancer screening. So I think it's going to be very important to prove value where you can derive an economic model to really make it work.
As a former commissioner on MedPAC, I might disagree with how hard, in what financial hardship Medicare has put the provider community up. But anyway, thank you. Anyone?
Well, I think Frederick makes good points. The economic value of using it is what reimbursement is set at. So there's been a lot of AI tools that are trying to replicate what people are doing. Those are not terribly valuable. Those are efficiency plays in dollar terms.
Exactly.
When you're impacting downstream care, then the health economics looks at that and says, we're saving unnecessary procedures. We're treating cancer early. It has clear benefits in the cost to treat in the end. And that's what we have to really look at. And that is not increasing the cost of healthcare by using the technology. It is intended to decrease the cost of care. And that's where you're seeing economic value and reimbursement being established for different technologies.
And to your point is, I'm not sure about the statistics, but I heard it's $230 billion that were spent on cancer care in the US last year. 3% were on preemptive care. If you look at the benefit for patient and even the cost, I love my pharma clients, of course, but do they really save patients' lives? You can ask yourself the question. So when you see the disparity between 3% in preemptive care versus 97% for treatment, it probably should be another balance.
Yeah, as this session is imaging biomarker and we have Optalum, so I want to say that Optalum has, we all know, successfully reimbursed in USA, right? But I think that for the economical perspective, we have to go furthermore. So they are doing some kind of a CADX program, detecting whether the nodule is malignant or not. But I think that we have to go further so that we can extract the physiologically make sense algorithm, or we have to develop some kind of biomarker that clinicians can understand. So I'm saying the white box biomarker, not just based on deep learning itself. So that I think that, and we think that that can be a way to be reimbursed for the lung cancer screening and help real clinicians as well as patients.
Rungyu makes a great point, and I think what he's saying is translating from the research side. As Ella said this morning, you start with the publications, the science, then you get to how to implement it into practice.
That's right.
And that reduction to practice is not by the number. It's not the number, the output of the CADX.
Yes.
It's what is your decision and how do you build the program? And that's why we're looking end to end at how to implement the program.
As I think Prevent Cancer Foundation is here, Bo Aldeje, and I think one of your statements is that the best cancer screening is the cancer screening that gets implemented.
Of AI today, and we, based on the experience that we have built now with the NHS in the UK, really supporting a near national program for screening. So we looked into the models. Where is real value? Are we trying, what is the return on investment for somebody who's paying the AI? And the business model today is per click per patient. And actually, AI proves really valuable at scale. So maybe for smaller institutions, it doesn't make too much, it doesn't bring too much value. So if you're trying to talk to at single center or single facility, it's very difficult to really come to a business model where it makes value, at least for today. maybe in the future, some of us are going to crack that egg, but today value is really shown at scale. So larger program, national program, where there you can hit the, either the shortage of radiologists or of trained radiologists. So yeah, for us, it's really AI has value at scale.
I want to thank the panel. It has been terrific, and I think we're out of time.
No, we have a little more time.
No?
We're gonna give it a few more because we started.
You don't want to keep the schedule?
No.
Oh.
I want to give this group a little more time. This is a really hot topic, and I think this group deserves to answer a few more questions. There are a lot of products out there now. I mean, there's I see people have an indeterminate nodule and then they go and get a nasal swab, or they're going and getting a blood test that's costing them $600. Three times the amount of the CT scan itself. Where is this heading? Immune markers, imaging markers, deep learning. Where are we heading with all this?
Okay. So there's a specific question of the cost effectiveness of the biomarkers, which I think they agree with that. I mean, to be implemented, to be reimbursed, has to be cost effective. And in fact, there are some models published on the price that could be acceptable for a specific biomarker, depending on the characteristics of biomarkers, and there are several qualifications. And it is possible to have biomarkers that are less costly than something that's question. The question is, which I ask myself sometimes, at least related to the previous question, the previous topic. AI is obviously coming up. And you just look at PubMed publications and AI and lung cancer screening or biomarker generates. It's incredible. Highly significant. But the idea of the white box AI, the idea of what the clinicians need. I think we were talking a lot say that clinicians need a score. Something very simple, 0 to 10, and that's it. And they need to understand what the score means. So the topic of the area of interest, for example, the utility of AI is a very hot topic also. So, and then the multimodal aspect of what we measure with AI. So it's kind of very complex issue. The multimodal meaning clinical data, biomarkers, liquid biopsy biomarkers, imaging, because we are talking about AI, but AI is not only for imaging. It also applies to liquid biopsy biomarkers or to clinical data. So this is extremely complex input that has to become a very simple output, cost effective. So it's a challenge. I agree.
Yeah, I mean, I could imagine somebody going for their nasal swab and getting one result, getting the image analysis to say another result. I mean, then getting a blood test and getting a third result.
If not three inputs, and three will one with the other. So then we, for the clinician, really something that assures him or her a lot.
But I think in reality, imaging will never go away. Imaging will win because it has a number of fundamental advantages compared to all this jungle of other molecular biomarkers. Well, it's more cost effective because of CT scan is already there. But importantly, it's instant. You have the result, the score 1 to 10 within seconds. You don't need to wait a couple of weeks to send the results to a central lab. And it provides anatomical localization to drive precision procedures like we've seen in the previous session on precision biopsies. imagine multifocal GGOs or lesions, certain regions very frequent. And I think the blood biomarkers can maybe complement imaging, but imaging will be driving the way.
It's been a long time since he did any work, isn't it?
Anyway, he always says morphology goes always after biology, after molecular changes. And he kept saying to me this is the very one of morphology, the imaging. There are aspects, obviously, in which imaging will be the solution of this area. But there are other aspects which the molecular data, but systemic biomarkers that you can find not in the lung or in the image, that you can find them 3 years in advance, 4 years in advance, because the molecular change has happened already in a very small nodule that probably imaging will not have the power. So that's a difference. I'm not saying, I think this is why I'm saying that all technologies need to be developed. And this morning, somebody was talking about research. We need to push to get more funding on research and in the different aspects, the imaging, the artificial intelligence, the blood-based biomarkers. By the way, there are many more blood-based biomarkers than ctDNA. There are a lot of them that are very, very interesting and very powerful and very robust. So it's not only ctDNA, but the idea is research. We have to and for example, if there is a screening cohort, it's very important at least to have the resources or to put the resources to keep the blood of these individuals in a pilot study. The Cassandra, for example, we are trying to find how to fund the blood collection of this cohort, because it's a pity if we spend 5 years doing a very difficult project, lots of images, we will get a lot of imaging information and radiomics, etc., but we lose a lot of opportunities. I think it might be.
See, I'm a very simple guy. I look at all these complex blood tests that can be done and all of this incredible AI or image analysis. But the thing that always is circulating in the back of my mind is measuring change in size is, to me, still the most powerful measure of, is something malignant or not? Is it growing? Are we growing at a malignant rate? And I always want to know that we're doing better than that. And that's always sort of my guiding light. And this simple concept has been around since the early 50s. Look for change in size. Now, as one sort of follow-up question for everybody in the imaging side, how confident are you about the robustness of your malignancy score or whatever you're producing, given slice thickness, newer scanners, 0.2 millimeter slice thickness versus 2.5 millimeter slice thickness, things like that. How good is your quality? And this is going to be a topic. I know we can't answer this all right now, so I'm not expecting it. But I know tomorrow we have a lot of sessions on this and about image quality. And this is something that concerns me in the back of my, it's not so much in the back, it's right in front. I'm looking for comments.
So I trusted with my own life. I have my own incidental nodule in a scan ordered by asthma doctor, and I did not go for a follow-up because my score was 1.
Good for you.
The thing you need to look at is that has to be open in the evidence. So you have to have generalizability. In order to prove generalizability, you have to test it in a lot of populations. And once those tests are done, then you move from research to practice.
Yeah. The idea, I think, of of how useful the products work. And all of you have really interesting products. I don't know which is better or worse or whatever. I'm sure they all perform at a pretty high level. But I'm very curious how different they'll perform if you had all your data from a 0.2 millimeter photon counting scanner versus, whatever, NLST data or something like that, which is, 10, 20 years old. So it's an interesting question. Your products will only get better. I mean, that's the one thing I'm positive of. This only goes in one direction, and that is getting better.
Yeah, to your point, I think we've hardly started to see the tip of the iceberg of what the technology can do. And I think we're going to be making more progress in the next 5 years than we've done in the past 100 years. And I think it's moving at such a pace now. So just like for any test, it's going to improve. Of course, over time, the more data, the more tests are done for robustness. But I would, follow close to your claim. I think it's already starting to become very, very robust. It will only get better. And what's being done for lung cancer will be done for liver cancer, for colorectal cancer, for all sorts of cancer. So I think we're just at the start of the way I see it, a major technology revolution, because if we talk about AI, that's technology. And that technology exists. And it's also new players coming in that had nothing to do with healthcare. The latest financial healthcare conference I went to present in Boston at a major financial healthcare conference. The star of the show was NVIDIA. And because NVIDIA was providing people like us with computing capacity, the GPUs, everybody had been using NVIDIA for a long time. And suddenly you have new players who come in. And I think this revolution will be hugely beneficial for everyone here.
Yeah, back to the first question, I would want to say that we have to I believe that clinicians should think technically and technicians should think clinically. So yeah, I think so. So for the malignancy score, we can say that it is a imaging biomarker, of course, yeah. But if we have to think clinically, so if you want to apply that score to the teenagers, which is absurd, teenagers have few rare lung cancer. So if you apply that model to teenagers, it will only generate false positives. But if you apply it to very high risk group, they will very beneficent. So I believe so my conclusion is that we have to think clinically, we have to think technically, both of them.
I have one last question, but I want to remind folks of the image that Dr. Yip put up this morning with the pirate ship. And I think that was an example of taking advantage of the capacity that Frederick mentioned. And by the way, I thought that was much better than the lousy poems that somebody read. But the my question is given the dramatically decreasing price of AI, what's that going to do again, kind of a business model, the thoughts about that and the price of the AI-based liquid or imaging add-ons?
I think in healthcare, like in pharmaceuticals, it costs nothing to produce a pill. It costs cents and it can sell for $50,000. So it's the regulation, FDA reimbursement approvals is what protects the value. I mean, healthcare is the only field where the cost of technology only goes up. Right? It's not ideal, but I don't think you see replication of like free technology because of the burden of clinical evidence and regulatory, which does not exist in other industries.
I necessarily don't agree, actually. So at a scale of operations, when you actually deploy to a lot more, the cost would significantly go lower because of the same product getting adopted at multiple sites. So it's an unanswered question, to be very honest, as how much, because as we keep deploying, we get newer nuances in data and we start implementing or churning newer things with respect to that data and trying to understand how to go about it. But if we understood that this is the stable product and this will reach it, this is the one that is to scale, you would reach a unit economics where it is going to go low. In terms of business model, I think typically AI companies operate on like 3 models. When you think about cost, when you think about pricing your AI model, it is like, hey, I'm giving you this product, here's the benefit. So the cost, the benefit is at a certain value. But because of false positives or other costs, there's a cost function as well. So playing with these two comes up with the what value you want to price your AI model. But you could also be a value-based care that is, I think, is growing topic in US. So value-based care, when I'm able to show that here's how we are proving advantage, or here's how we are improving patient outcome, functional outcomes, operational outcomes to a site, that can also lead to a cost model that would make sense for the sites. Yeah.
I think we're out of time.
Yes.
I want to thank the panelists. This was a wonderful discussion. I wish you all good luck with the development of your products. I'm sure we're going to see wonderful things in the near future. And keep up the hard work.
Thank you for the
Thank you for the
opportunity.
invitation.
Very good. Very good.
And now, Dr. Henschke, you are. I'll introduce Dr. Henschke, who's going to introduce Dr. Myers.